Welcome, everyone, and thank you for joining today's webinar, Agility with Complex Datasets, Integration of Predicted and Empirical Data for Rapid Innovation in Drug Discovery. Before we begin, a few brief housekeeping items. The session will run for approximately 60 minutes, including a 15-minute Q&A period at the end. We encourage you to submit your questions throughout the presentation using the Q&A tab at the bottom of your screen. We will address as many questions as possible during the dedicated Q&A session, and today's session will be recorded. All registered participants will receive access to the recording via email within 24 hours after the webinar. Today's discussion focuses on utilizing complex datasets in an agile manner in the context of protein dynamics in drug discovery. Our speakers from Congruence will lay out the current landscape and the challenges in the field, discuss how a component of Congruence's drug discovery engine, Revenir Knowledge Base, RVKB, streamlines the integration of chemistry, biology, and computational data. With that, I'm pleased to introduce our distinguished speakers from Congruence Therapeutics. First, we have Dr. Maximilian Ebert. He's Executive Director of Computational Chemistry at Congruence, and he brings extensive experience in molecular dynamics and free energy calculations. His previous work developing the Molecular Operating Environment platform has been instrumental in advancing computational approaches in drug discovery. Next, we have Dr. Jeremy DePaul-Chiquan. He is Associate Director of Biology at Congruence and currently leads the GBA1 allosteric modulator program, bringing nearly a decade of drug discovery experience to our discussion. And last but certainly not least, we have Dr. Lee Fader, Vice President of Chemistry at Congruence. His background is in small molecule discovery, which also includes leadership roles at Ventus Therapeutics and Inception Sciences before Congruence. Thank you, Lee, Jeremy, and Max, for joining me and the audience today to chat about harnessing proteins for drug discovery. Let's first give a bit of background on what the protein therapeutics landscape currently looks like, current challenges, how Congruence fits in, what the future looks like, and a little taste of what the day-to-day kind of looks like. Does that sound good? Let's get into it. Lee, over to you.
Great. Thank you very much, Josh, for the invitation to participate and also the kind introductions. This will be a conversation with Max, Jeremy, and I about a little bit about Congruence, but I think we're going to touch on a lot of complex issues that many people deal with in our industry, especially in the sphere of biotech and companies that leverage power of different types of platforms that generate these complex data sets. So what I'll do is from a very high level, set the stage by telling you a little bit about Congruence. I have a slide here, which I don't want it to feel too corporate, but I just want to give you an idea about where we sit in the landscape of different companies. We're a computationally driven biotech company with a proprietary pipeline of compounds headed toward the clinic. We've got one program in the lead, and we anticipate initiation of phase 1 study of this molecule, and the nomination of DCs in the other 2 projects, and all of this over the course of the next 18 months or so. Our pipeline includes projects directed toward helping patients afflicted by MC4R deficient genetic obesity, GBA-driven Parkinson's disease, and alpha-1 antitrypsin deficiency. Like I said, we're leveraging our proprietary computational platform, Revenir. Max will talk a lot about that, but essentially we're capturing protein dynamics in a very unique way in order to recapitulate disease caused by, in the earliest cases at the beginning, due to misfolding of proteins, and we predict the correction of these defects by small molecules. And we are, a well-capitalized company with the resources needed to drive these programs forward. So I'll hand it to Max. He'll pick up for a few slides, and then we'll bat it around a little bit and discuss this complex issue.
All right, thanks, Lee. So At Congruence, we are developing a computational platform for identifying small molecules binding to various different targets. We call it revenir, which is French for coming back. And our current focus is in the discovery of pharmacological correctors or chaperones. It's a large and well-defined opportunity. Protein instability is a key defect in large number of diseases, rare and monogenetic. And this instability leads to loss of function through protein degradation, mistrafficking, or even gain of toxic function like aggregation. And you see an example here on the right, where we see the wild type protein is well folded in its native state, and then we have a perturbation. And this is oftentimes caused by a pathogenic mutation. You have a conformational change that leads to a less stable protein. And now all of a sudden we have a loss of function and gain of toxic function. And what the Revenir platform is designed for is to identify binding pockets for small molecule stabilizers, which then correct the misfolded protein, improve trafficking, restore function, and prevent aggregation. This has been shown to be a commercial success with Trikafta from Vertex or Vyndaqel from Pfizer. And what we try at Congruence with the Revenir platform is to really dramatically improve the traditional discovery methods to efficient and increase the probability of success versus traditional means like high throughput screening. And our platform was internally validated extensively, and we've shown internally multiple times that we can rediscover these pharmacological correctors from the literature and historic projects. So on the next slide, what I wanted to show here is a basic high-level overview on how the platform works and how do we achieve identifying not only small molecules in terms of target engagement to the target, but to predict biological function. So it's a four-step process. And in step one, which we call reveal, is we reveal the molecular basis of the disease by modeling the hidden features in protein dynamic ensembles. And essentially, we are generating for the wild type or reference state a conformation ensemble, because proteins are not static in nature. Their native state is dynamic. And so the entire platform is based on protein ensemble calculations. And then we also generate the pathogenic variant and an ensemble and create a sophisticated machine learning model, which is able to differentiate between the benign variants and the wild type, and what is different in a pathogenic variant. When we have this disease model, we further leverage the protein dynamic ensemble in step two, which we call dynamic pocket hunting. And the goal here is to discover allosteric sites and cryptic pockets for their propensity for ligand binding, and also for the potential to revert the disease state. And that's very important because in protein ensembles, we often see many different sites appear. And the Revenir platform has components to rank order these different binding pockets, not only for the propensity for ligand binding, but also for their potential to be an allosteric modification site. Once we have prioritized the sites, we are building ensemble-based pharmacophore hypotheses for screening large virtual high-throughput screening databases, which we curate internally, or leverage tools such as machine learning assisted docking to assess billions of molecules, or free energy calculations, all with the goal to get a hypothesis for target engagement in these pockets. But for correctors and chaperones, that's not all you need. We don't need only binding to our target. We also need to revert the disease state. We need to correct for the disease introduced by the pathogenic variant. And that brings us to step three, and we think it's very unique to our company. And we call this achieving congruence, where we apply real-time ligand-based correction to predict if the small molecule hit has the power to resolve the disease state. So essentially, our top 100 or 1,000 hits from the target engagement in step two are then put into protein dynamics simulations again, where we have the pathogenic variant in context of the potential corrector. And then we see, based on the machine learning model from step 1, is now the ensemble of the pathogenic variant more like the benign or wild type reference conformation ensemble, or do we stay in the disease conformations? And so this allows us to rank order these compounds not only based on the prediction that they engage with the target, but also for the biological function. And so these high-value hits are then passed on to step 4, which is testing where these compounds then progress from the dry lab to the wet lab for lead generation.
Yeah. Excellent. So I think, at this point, there are a couple different ways where we could sort of continue to highlight where the complexity comes in. But I think what's clear, Max, and you can back me up on this, is this process, no matter how you do it, if you imagine looking at ensembles and ligand binding and comparing ensembles to each other, what you're generating is a lot of complex information. Do you more or less agree with that?
Yeah, sure. And I think one of the key aspects today is to kind of show you a little bit on how we not only store, but manage and share the information from the computational platform, but marry this with all the results coming in from biology, pharmacology, and chemistry.
Right. So once we, pass these hits and have identified hits into the wet lab, a lot of people will be familiar with a central paradigm that looks something like this. We often talk about this in our line of work, and medicinal chemists especially will be very familiar with this sort of design, make, test, and analyze cycle. I want to walk through it a little bit just to set the stage a little further for the problem Max has started to illuminate. We know that let's enter the cycle at the test stage here, because that's sort of where Max left off. We will take a list of molecules that may have come from different sources. We may have synthesized them. We may have purchased them. We test them in assays that are designed to tell us about will our compounds be efficacious? Will our compounds be drug-like and have a suitable pharmacokinetic profile or even safety profile, things like that. And then at the end of this, we analyze this data. We generate new hypotheses about what molecules we should make next, or what kind of profile we need, or how we can fix this. This analysis can involve predictive models. It can involve traditional approaches to examining SAR, things like that. But at the end of the day, again, we generate a lot of complex information. We then take that information and insert it into our next design cycle. And that is, again, where we will use perspective modeling, different approaches to selecting different molecules. And what I like to do, and I think a lot of people do this, a lot of companies have this sort of culture, is to capture that those design decisions. And so that's another type of data that we endeavor to capture. And then, of course, we need to effectively deploy resources to make the next round of compounds. And there's a lot of complex data that's generated. And so I think what we're talking about today is how do we warehouse all of this different type of data effectively. And this cycle for us includes use of our platform. So every time we design new molecules, we use the platform in order to help us select targets to make next. So we're constantly generating these complex data sets that have predicted values in them. And
you bring up a really good point, because every computational platform obviously improves when you have access to actual wet lab data. And so our paradigm at Congruence is really to be as closely connected to all the functions in biology, chemistry, and computational chemistry, so that the platform can be informed by the results, but also suggest what to do next. So in some cases, we have a gap in our models, and while the compounds might not immediately be valuable for the specific program, but these compounds might help to make better predictive models moving forward. So there is an intrinsic cycle where we go from improving the platform and prediction, and then generating the next round of compounds, and having access and be basically fully integrated into the wet lab has a tremendous value to also move the platform further, because like every software, and we know that from CDD Vault as well, right? Every software is constantly evolving, and you're never done. And so being so well integrated has a tremendous value. We see that here at Congruence.
Yeah, 100% agree, Max. That's a great point. I was going to then, kind of highlight one of, in the way that made sense to me, at least, how this data flows, where we keep it, and give it a little bit of an introduction to, how we deal with all of the information. So the top part of the graph is really about how, we're a small biotech. We interact with a lot of third-party organizations that help us execute on our research plans. So we have the typical contract research organizations who might do chemical synthesis. They may run experiments in biology and pharmacology. We generate a lot of data with these partners, generate a lot of value, a lot of data, and then what we selected as a small company who needed a reliable solution to data house warehousing is we selected CDD Vault to really hold the data. I won't do the pitch for CDD Vault, why it's as great as it is. I'll let you guys handle that. But I do think it's an excellent solution, and has been an excellent solution for us. We also have different types of partners that I'm just calling vendors and service providers that generate different types of data that we also need to warehouse. And then, of course, critical to us is the work done in our biology group and pharmacology group in-house, that it generates very complex data in our own labs that we also connect and store, depending on the data, connect and store to CDD Vault. So this is wet lab complex data coming together that we need to, connect to the predictive data that Max's group is using. And so, what we do is have a line of communication between CDD Vault and what we call the Revenir knowledge base, or we'll call it RVKB for today. And this can have a lot of different types of data. Some of it is processed versions of what's in CDD Vault. Some of it is predictions, Revenir predictions, or sophisticated ML predictions. And sometimes what we do is further process that data or aggregate it in different ways, and even model it in some ways using a complementary tool called KNIME, and this is all brokered by the KNIME Business Hub, which I won't talk too much about. But we sometimes use KNIME Hub to help with the speed of the communication between CDD Vault and RVKB, just because it's a little slower to rely on REST APIs directly to CDD Vault for applications that require, essentially live refresh of data and things like that. Then we have a design suite that's connected to RVKB and CDD Vault. That will feature a lot of different tools and platforms and approaches. I name a few here. Revenir is involved. We use Mo typically for modeling, KNIME for a lot of cheminformatic tasks and visualization software like Spotfire and others. And then, of course, on the far left, we have custom front ends in order to do some visualization, some data analysis, and present data to different stakeholders for consumption and for different purposes. So all of this is just wet lab communication to the central data warehouse and flow to the central warehouse. The key box at the bottom, the RVKB box, is also a node where all the computational work is fed into that. I think Max will pick up in the next slides and talk a little bit about the complexity there.
So, I mean, let's talk about the complexity
Yeah.
and basically.
You want to stay here, Max, or do you want
No,
me to
just
move ahead?
go ahead, because I wanted to touch a bit on why do we saw the need for a knowledge base. And a lot of software exists to manage internal data, but there is obviously a lot of value in external data, especially for computational team. But also for every other branch. You know, you don't want to fly blind in your drug discovery process. You want to have the bigger picture view on your project. And there's a lot of importance in bringing this external data in-house. And one of the key factors is data safety and security. I mean, we probably have all been there. There's a great web service for maybe a property prediction or a retrosynthesis analysis. But because it's a public web service, we can't just copy paste our own proprietary smiles in there. We want to be in control of where our data goes and who is actually processing it. So sensitive data should always stay inside, but it's challenging because there's a lot of useful tools out there. So we thought we need a knowledge base, which is just an overarching term for bringing services and data to the researchers here at Congruence. But there's another problem with bringing these interfaces to people is that everyone has their own paradigm, their own way of interacting with chemistry or biology. So there's a steep learning curve for new team members if they have to learn like 10, 15 web services, and so knowledge transfer is difficult. And so having a centralized data infrastructure like the Ravinian knowledge base provides a consistent and accessible interface across the organization. And it also allows people which are not on the day-to-day data processing and generation to see what is going on. And I'm thinking there about the administration, the executive members of a company, and they also might want to know a little bit about the current progress, and having a direct access in a very short communication chain to the most recent progress in the company is very, very helpful. And so this streamlined access and reliability really helps us to be ready for the years to come. In the past, I wrote a current opinion a long time ago during my PhD, and it was looking and analyzing a computational algorithms and services which were published and made available either on GitHub, GitLab, or just as a web service. And a vast majority of these tools which were published and in literature were not accessible just 3 years after the publication, because maybe the professor had moved on to a new institution and didn't bring over the web server, or it was not functional anymore because there was a new recent update to Windows, and etc. So bringing in these really essential tools and making them available for the long term is very important when they become a key piece of the drug discovery cycle. So have a reliable access and reduce dependency on these external platforms has a huge value. But obviously that comes with challenges, right? So we have this external data integration battle with non-uniform data formats. Many of us might have visited PubChem, NCBI, the RCSB, etc. And everyone has their own way of sharing and compute and providing access to data. And also, so integrating these data is difficult, and we'll come back to how we do that a bit later. But another problem is also the access speed. So sometimes the server might be down, but then you have a critical timeline in your company, and how do you deal with that? And also, the speed might be slow. You will have requests like multiple per second, and then you will be blocked because of the protection of their own web server. So all that is problematic if it becomes mission critical for the company. And then there's also validation. If the data is constantly changing in these sources, how do you deal with reliability? And we wanted to solve that by having the knowledge base. And finally, there's also the complexity of unstructured data. Very, very valuable data is in publications, get it from conferences, and then often in non-machine readable formats. So we want to digitize that and keep it in the records and access it from our fingertips. So that was the reason why we thought a knowledge base is very valuable for us moving forward. And if you could just go to the next slide, I just want to show like a little bit of a glimpse of this data broker, a middleware solution, which is bridging the gap between all the disciplines at Congruence. We have two key features. We have a front end where I put some screenshots here where we can query for molecules and structures and mutations we've analyzed, find docking scores or other computational prediction like molecular dynamics simulations, but also all the wet lab data. But then what is more important for us is the robust back end with the exchange framework we put in place, which harmonizes all the diverse data we generate from every corner of the company, and then have a consistent communication to provide the data where the actual researcher is. In the biology ELN, and Jeremy will give you an example of that a bit later. For the chemists, obviously in CDD Vault, and everyone should have access to the data so that you basically empower the decision makers. It's not about replacing anyone, but it's really empowering everyone to have access to the data, to the most recent findings, instead of waiting for the project update. We've all been in the meetings. There's a PowerPoint presentation. We get the update. We get new ideas, but we want this creativity to flow constantly at Congruence, so everyone has access to everything at all times. That is the overarching goal here. So it facilitates obviously the collaboration across our functions and makes the data accessible as well. And I thought I'd give you one example of that on the next slide. And what happens when at Congruence a chemist comes up with a new idea and deposits a molecule in CDD Vault. So this chemistry can obviously come from various sources. It could be a screening result from the Rivernear platform, a new design idea from our med chemist, or it could be a molecule from our GenAI platform where we made compounds tailor-made for our identified pockets and pharmacophore hypothesis. But in the end, we have a molecule which we deposit in the CDD Vault, and we are all familiar with this process. But then our knowledge base is retrieving every 30 minutes new compounds deposited. And what then happens is that we stereo enumerate the compounds if necessary, assign a series label to it. We've identified which pHs are important for the project, then prepare the compounds at the correct pH, assign it an internal identifier in the knowledge base, and store these compounds in the in-house databases. And then we send out an email because that all happens in the background automatically through data pipelines, which I will discuss a bit later, and we can dive into that a bit more. But then an email is being sent out to the computational chemists, making them aware that there's new data available or new compound ideas available for a specific project. We automatically also run prediction in machine learning models and AI models, and then feed this information back into where the chemists are, obviously in CDD Vault. But the computational chemist will then take these medicinal chemist ideas and pass it through the Revenir discovery cycle. And Lee and I were briefly talking about that earlier, because these design ideas are immediately evaluated for their potential for target engagement, but also to predict the functional rescue potential for our targets, which we are currently having in our pipeline. This is all informed by public data, but then we also wanted the biologists to be aware right away of the new chemistry, and if these were already made, which batch we are currently operating on, properties for these compounds like molecular weight, TPSA, or just activity model data. We often establish also a special lingo in the company, right? So we also share what synonyms are used within a company. And so all that is done automatically in the background so that everyone has constantly access to all the data. And how is that achieved? So if we just move one further, so essentially we have the data pipeline architecture. It's often quite easy to get the initial data, especially when it's from external sources. We can download it, but then it's on the computer of someone, and it never gets updated. So we basically create data input pipelines, which facilitate the integration of these diverse data sets for the most important sources. But we also make it updatable. So we ensure that we have a current data when we think about, let's say, the RCSB, that we have constantly access to the most recent new structures which are deposited. So we have automatic checks and validation for accuracy built in. And then we build also output pipelines, right? Because we want to then distribute this information into the different functions at Congruence. And this also, and this is something that we recently added to the knowledge base, includes searchability of unstructured data. And with the advent of the large language models, and we have probably all use ChatGPT, we essentially made Google Gemini's platform an intrinsic part here to be able to search for certain internal numbers, or just with normal natural language ask questions, and the response will be based on our PowerPoints, Word documents, or lab notes, and so that makes it really easy to quickly access data across. I mean, there are obviously advantages. It reduces dependency on external formats. It lowers the learning curve for new employees, as I mentioned before, and just enhances the decision-making process, because when you have the full bigger picture of what is going on in all the projects internally and externally, it improves the accuracy and the speed at which the decision-making process is happening. And one corner piece of that is obviously machine learning models. And this is where the knowledge base approach really thrives. So when on the next page, we have our MLOps paradigm, which we call internal property model. It's a piece of software which really thrives in this environment where data is accessible and up to date at all time, because we can do training, deployment, and consumption, as well as validation in a constant cycle. And we really live this constant update of models, because we have this centralized data store and can provide these experimental observables to our pipeline, so that we can constantly update the machine learning algorithms we use. We do work a lot with information gain technology. So that's what I meant when compounds are suggested to make to improve the model accuracy. So we really like to double down on this continuous model improvement. And while I think a lot of times our field or my field has like overpromised and underdelivered, I think we at Congruence we strike a great balance between using novel state-of-the-art technologies, but then rely as well on well-established paradigms in the drug discovery cycle. So I think it would be a great time for my colleagues to chime in and maybe look at how we use also tools like KNIME treat data and trigger, let's say, different processes.
Yeah, 100%, Max. Thanks for going through that. I think, for me, as a chemist, it's easy to imagine that a lot of what you've just talked about is integrated into different steps of our process. What I had in mind was to present one of them. There's some animation, but I don't want here. And that is how we, use a lot of the different tools that are developed in our design process and how we manage the information that's generated. Because at the end of the day, we use tools like Reveneer, like models, ML models for properties or potency or different things like that. Even sometimes external tools that we haven't developed, but we've integrated into our workflow, and how we use that information to drive decisions is something that we have tried to capture essentially in our process. And the real reason to do that for me is to generate the indicators that were maybe slipping on our cycle times. That's, I think, something that chemists are always focused on. The worst thing you can do is generate data and not respond to it and not use it to design the next round of compounds. And part of our endeavor, I showed that cycle at the beginning, the design, make, test, analyze cycle, as the way I call it. This is an iterative process that we don't do once. We do it many, many times. And our goal is to do each cycle as fast as we can, but also do as few cycles as we possibly can. So what I'm showing here is how we manage our design process. What we've essentially done is taken an additional vault in CDD and used it as our virtual playground, essentially, to enumerate ideas, capture decisions, capture hypotheses. And I'm showing some of that in this table in the molecule fields and the batch fields in our CDD sandbox vault. So this is not a real example from Congruence. It's a literature compound that maybe is close to my heart and some of my former colleagues will recognize. But I've just added it to our vault to show, to give an example of how we do things and manage information. So basically, you can imagine when you're designing new molecules, you're doing this in a lot of different ways. We have design sessions. We meet with our consultants. We get together as a department and brainstorm things like that. And so what we endeavor to do is capture all of these ideas, whether we're going to make them or not, in our sandbox vault. And that really enables Max's team to then pull the structures out, run the predictions, and provide the predictions that will help us prioritize targets. And then what we do is we use the sandbox vault with our vendors, our chemists at CROs to essentially publish to them our prioritized lists of compounds to make, things like that. If we make changes to priority, we annotate it here, and that in turn informs our partners what should be next in the queue, things like that. We can also deprioritize things and all of those usual operations, but we capture it all here and timestamp it here in CDD Vault. when a change is made, such as, when a compound has been made, what we do is as part of the sort of upload of new information to the batch fields from the CRO, we give changes to status. And when that status is changed to ship, as I've shown here, there's a trigger that occurs, sort of brokered by the KNIME Business Hub that takes that molecule with all its analytical data and information and registers it into our actual registration vault and tees it up for actual empirical testing. And so here we can sort of manage the conception to synthesis to testing part of the cycle time really well with very high fidelity. And this is it's really these two tools working really well together that enable us to do this. So that's one other part of our data flow that we thought might be of interest to people. The next slide, I think, is where we take that step from we've made a molecule and what do we do next? So I'll just switch over to this slide and pass the baton.
Yeah, thanks, Lee. So here's an example of RVKB and how we use it in-house for biology. So like Max mentioned, the information as soon as the molecule is synthesized and in the CTP vault and then uploaded on RVKB, but also on our biology ELN. So on the left-hand side, you have a snapshot of the same molecule that Lee was showing on the previous slide, where it has all of the basic information for us, including, let's say, the molecular weight. But it would also contain all of the external data sets that would have been produced either in congruence or by external partners. And this same set of information regarding the molecule is also uploaded on our biology ELNs. And so this allows our scientists to tag the molecules as they're doing the experiments. It allows us to track which experiments are being done with which batch of molecule. And all of this is seamless, right? So we don't, as soon as a molecule is being synthesized, we can easily track this information. And then on the other side, RVKB, what it allows, let's say, scientists to do is he can then go and check, what else has been done with this molecule, how does this profile look like, and if he's curious about, the docking poses, then he can go and have access to this information just on a centralized RVKB. So this is all great and allows for data harmonization and transparency across all of the different departments. So if we go to the next slide, I think it's again reiterating, why we're trying to harmonize data, right? So I think we're all aware that, drug discovery can't be really done in a silo. So everybody needs to be able to interact with each other and see the data. There's chemists, biology, pharmacology, and computational. We all try to come together, but we're also all generating distinct data types, right? So this leads to different types of challenges. So including, let's say, data storage. So for chemistry and biology, I think there's good software out there. And like we showed you here, we were able to interlink them. So I think Max on the next slide is going to go through some of the problems that maybe computational scientists have.
I just wanted to really briefly touch on this topic before we end and go on to a question. So if we just move ahead, like Jeremy was saying, there are great solutions for chemistry and biology, but for computational chemistry, it's more difficult. The challenges like molecular dynamic simulation, they generate terabytes of data in different formats, proprietary formats. They're binary, non-transportable. They often require proprietary software to read it. So there's just a problem in organizing and make it trackable. There's also a lack of standard protocols. Oftentimes, models are inaccurate. We need to often change the models and the methods depending on the current task at hand. So the result is that computation chemistry really lacks a unified system for managing, sharing, and documenting project data. And we just go ahead one slide. I mean, there's some tools. I mean, every company or group will use different a set of tools, but some of the current options would be just documenting lab notes on OneNote or use Jupyter notebooks. But while they're user-friendly for computation chemists and versatile, they're not so much for like our colleagues in biology. Like, how do they interpret it? They, getting or running a Jupyter notebook on a regular machine, lots of complexity involved with that, and the same as how we store code and data in GitLab, but for then storing also the large training set data, Git is not the right the right environment. And so the specialized data management systems, they primarily focus on experimental data, but computational data, it's not covered that much. And even if you can store it, then there's less tools for even interpreting the data. So this is a limitation, and we tried with the knowledge base to overcome that here at Congruence. And if we just go to the last slide to kind of conclude that, and as a future outlook, if there's just one takeaway from our discussion around here, is that integrating the data, documenting it, sharing it, can really elevate our contributions to drug discovery. I mean, here at Congruence, our future goals is to enhance the knowledge base even further, and bring in even more data, share, and be more open and lower barriers. And these are all parts of our standardization efforts. So if there's a little bit of a call to action, is to embrace tools, really be collaborative, and develop practices that foster this data transparency and compatibility across all the scientific disciplines. And so this would be my conclusion. Joshua, maybe if you have a question, or we go move on to the question period.
Yeah, sounds great. Thank you. Thank you, Max. And thank you, Lee, and thank you, Jeremy, for that fantastic conversation. Now it's the time we'll move on to Q&A. All right. Well, thank you so much to the audience, and of course our great speakers for that the discussion right there. Before we launch into the questions, and we've gotten quite a few questions from the audience, thank you for your engagement. We'd like to just quickly throw up a poll for y'all. If you like what you saw, if you're curious about learning more about Congruence or CDD, or maybe even seeing a demo, please vote on this poll, and we'll be sure to have a member of our team reach out to you to start that conversation. Okay, I'll leave that up, and let's dive into the questions. If you haven't had a chance to submit any burning questions you have, feel free to please submit the questions via the Q&A tab at the bottom of your screen, via the control toolbar. Without further ado, let's get into it. The first question we have is from an anonymous attendee, and is asking, what kind of experimental data do you have on misfolded protein conformation? Jeremy, as the Associate Director of Biology at Congruence, maybe I'll popcorn this question to you first, if you don't mind.
Right. Thanks, Josh. Yeah, so obviously, we scatter through the literature to find as much information as possible. And then we have our own experimental data set in biology. But I'll also put Max in the loop here, because they also use quite a bit of information to try to model how these mutations affect the conformation of proteins.
Yeah, that's right. So obviously for protein misfolding, the actual experimental data is a bit sparse, and especially the direct structural data. There is cryo-EM and MR data and X-ray, obviously, available, but as we speak about misfolded proteins, they can't be structurally determined in many cases. So since we are a computational company, we generate our own physics-based data. And so we have developed internally an accelerated conformational generator, which we validated extensively against the available structural data, but also since, like I said, of the absence of real structural data for misfolded proteins. We also use HDX data to see which residues are more exposed upon mutation, and hence can we reflect that and see it and observe it in our computational ensemble. So it's a mixture of both, but in this particular field, we heavily rely on computationally generated ensemble data.
Awesome. Very cool. Thank you, Max and Jeremy. Appreciate that. Awesome. And so maybe continuing on, there's another kind of follow-up question that might be very similar, is or along the same lines, and it's asked by how do you computationally sample some of these hidden binding sites? Is there a certain kind of process or methodology you use?
Yeah, I think that's also probably in my realm. So a lot of time, these hidden binding sites or cryptic pockets are the hidden gems in drug discovery. And repeatedly, we've seen that in crystal structures or static structures, these sites do not appear. well, we can make them appear in conformational protein ensembles. Either this can be done classically with molecular dynamics, but then timescales are very, very long. And in our case, when we look at multiple mutations and maybe also effects on small molecules on these ensembles, we can't assess these long timescale molecular dynamics. So we, as I said before, we developed an in-house method for accelerated molecular dynamics, which is a hybrid method between elastic networks and all-atomistic molecular dynamic simulations. And this allows us to reveal these hidden pockets and hunt for novel binding sites in the conformational ensemble.
Awesome. Awesome. Very cool. Very cool. Thank you, Max. Appreciate you sharing that. Lee, maybe a question for you here is, since we are using CDD with multiple CROs, what are kind of the security processes that you've implemented to try to make things work amongst these different vendors?
Yeah, that's a great question. I'll give you the non-technical answer, I guess is the best way I can go, because I think our colleagues at CDD Vault are far better equipped to answer the question. But basically what we do is we rely on, the security that's built into CDD Vault and use CDD Vault itself as the entry point for the data that flows from the CROs in. So they have accounts, they're only exposed to the projects they work on. They're not exposed to, our entire database or anything like that. We also, use tricks like our projects are codenamed and things like that, just to keep things as anonymous as possible. But I think that the platform is incredibly secure, and we don't usually we don't have any concerns about using CDD Vault with external parties.
Thank you, Lee. I appreciate that feedback. Yeah, it's always great to hear. Mike Bauer is curious, what kind of pKa predictor are you using?
Well, I mean, that's a very good question. And we scratch our head around that a lot, because this is not an easy task. We explored neural networks which were published. We explored other methods with lookup tables or semi-empirical QM methods, which are provided by tools like MOE from CCG. And in the end, there's no perfect solution, which always works, but we have a generalized workhorse here for pKa prediction, which are the semi-empirical QM methods, which give us the best results across the board. But yeah, it would be good to have one tool which works for all the chemistry, but then for our series at Congruence, that is the best approach.
Totally. Makes sense. So thank you. Mike also has another question, but along different lines, and he's wondering if you've ever had any issues with your LLM hallucinating or anything like that.
Or maybe, Jeremy, you can start a bit, because in the biology group, we had a lot of discussions on that, and then I will follow up.
Yeah. So we do see hallucinations sometimes. I mean, so we use, I guess, two models, right? We use the generic ChatGPT, where we use, large public information to try to, gather, I guess, surface level kind of information. And internally, we have our own large language model, which we feed our own data to try to, find information more rapidly when you're looking for something maybe you've done a couple of months ago. But yeah, so I guess we do see hallucination, and it's something that we're actively working on. I guess, Max, maybe you can.
Yeah, I mean, for especially hallucination is occurring when we look at large language models which are publicly available. It will fill in some gaps and just invent facts, but it's very useful for getting initial access to large bodies of information. When we dive into a new project, we can just maybe upload the most recent 1,000 publications, get like a review article out of it, and it points us towards where to actually dive deep and go the classical route and read the scientific research articles. And to overcome this problem for our internal data, yeah, we train a blank LLM only on what we generate, what we communicate, and this allows us to quickly find information within the company without hallucination in most cases.
Gotcha. Very, very various approaches. I love that. And kind of along that line of thought, what kind of general best practices, themes are top of mind when you integrate, when you think about integrating AI and predicting kind of these drug pipelines and whatnot? What are kind of maybe the thought, like the themes, that are most top of mind right now, and whatnot? Yeah, and moving forward, of course, as well.
Yeah, sure. I mean, we use it obviously for a lot for property predictions and in the machine learning space. But before, oftentimes we usually call this QSAR modeling, and now it's a little bit more advanced. But at the foundation, it's still that, and it's most useful for the experimental scientists and the companies. So I had a slide on our internal property model, or IPM, which uses continuous integration and deployment to go rapidly from the data the scientists are depositing in CDD Vault, creating a model and then making it available, because if this runs on the laptop of a computational scientist, it's not really useful, and then you have to email and you have to wait. And so internally we have deployed either through a REST API or a web interface, or for our computational structural modeling tools, a way to tap into these models and basically access the properties where the scientists are, so that there's no middleman in the way. And so that's how we see it really useful, because it allows the actual scientists who are day-to-day working with these values and with the chemistry to kind of put us as in the computation team in check and see are the predictions really useful, or are they just generating great numbers, but in the end it doesn't move the project forward.
Makes sense. Gotcha. Thank you for that, Max. Appreciate that. Just let the audience know we are getting close to the end of the webinar, so this is your last chance. Please get those burning questions in for our amazing congruent speakers here. And if we don't get to all the questions during the webinar, we can follow up with them in an email afterwards as well. All right. So last few questions. I know we've been going rapid fire here, but please bear with me. This one's a bit of a mouthful, so if you'd like to refer to the Q&A tab, feel free to check there. Asked by Asma, if I understand this correctly, compound synthesis requests can be made to the synthesis CRO through Sandbox Vault, and once the compound gets synthesized/shipped, the compound gets automatically registered in the registration vault, correct? What about the specific custom fields defined by a company? Does that also automatically get updated when synthesis CRO share the information regarding synthesized compounds, or do such fields have to be filed manually? So that's like a three-part question. And
but the first thing to say out of the gate is that I do think that you understand the process pretty well. The thing to remember too is the passage of the information from the sandbox to the registration vault is brokered by the KNIME hub. So that's a commercial piece of software that it's sort of a server that that you do have to pay for. But if you're using KNIME, you can build the workflows to do the transfer of information from your desktop application, and it works great. Honestly, this is how everything is built and tested before it's deployed to the business hub. But I will say that with reference to specialized fields and things like that, the sky is really the limit. It's up to you what you put in and how you use each of these fields in the sandbox. The sandbox is really for us this a virtual compound collection until things are made. And so the way we do it, as you would normally do a batch upload of compounds, let's say, our partners, they upload batch information to the sandbox, and then the precise information that we want to be represented in the actual registration vault is migrated. This is all done, answering a previous question, this is all done with the REST API that CDD Vault has. They've got really great documentation on it. It's easy to use with KNIME and any way you would use a REST API, but within KNIME, it's very simple, and you're quite easily able to build these workflows that pass the information with high fidelity. So I recommend giving it a try.
Thank you, Lee. Appreciate you for tackling that one.
Yeah, there was another question I thought about. No, maybe I missed it. I'll follow up after, but I thought there was another part to that question. Let's forget it.
Okay. We have one more pretty interesting question, I'd say, from Lillian, and she's asking, if your data infrastructure got completely wiped, how would you start from scratch again? Were there any painful lessons learned? Maybe we'll kind of go around with it. I'd love to hear everyone's opinion on this, everyone's thoughts on this. Maybe Lee, start with you.
Yeah, I'm gonna start because Max is gonna give the real answer, so he should probably finish. But the first thing is all of our wet lab data, all of our compound information data, the real source, the place where that information is stored is in CDD Vault. So CDD Vault takes care of backups and all of that kind of stuff. We basically never worry about anything happening to that data. Anything we add that isn't wet lab data or compound data is also maintained really well. But then speaking more about the entire infrastructure of our RVKB, I'll definitely pass that to Max.
All right, yeah. I mean, we obviously have multiple backups. In the end, everything is backed up in AWS Glacier, where we store the raw format of our RVKB, but then there's also the actual compute infrastructure, our machine or MLOps and DevOps infrastructure, which we abstracted away as infrastructure as code. So we can very quickly redeploy the entire infrastructure, including our compute clusters in the cloud, either AWS or Google Cloud, because everything is written in CloudFormation or in Terraform. So we hope that we will never get to this critical moment, but I think we've all been burned by data loss in any shape or form, and that is why we have these strategies, have the raw data backed up in multiple places. I think many companies have that, and for the actual IT infrastructure, it is fully written as infrastructure as code.
Awesome. Wow. That was a heavy hitter question. That was a heavy hitter answer right there. Love it. Jeremy, any last thoughts to add on that, or should we move on?
Yeah, I think also maybe to go back to the question, I think it's what would you do differently, Max? So so my job is really to communicate what the needs of the biology team are and try to implement all of this and see how that works. But if you had to start from scratch, Max, like, would you do anything differently? Would you want different programs to be used, or right?
Yeah, I mean, this is a very long question, and maybe to give a short answer, I mean, the fewer tools you can use, the better, because a lot of time is spent by harmonizing the data, writing pipelines, and everywhere things can break and data can get lost. So if there would be a one fits all platform, that would be probably the best on that.
Sounds good. We'll taking notes here on the side here. I think with thank you so much for your time. I think we'll wrap up with this last final question. A lot of some of the attendees, really love the presentation. So thank you so much for your time. And some identified that this presentation really focused on the data flow and data management aspect of things. And they're curious to hear if you have any plans on presenting maybe your computational approaches or sharing more of what's behind the curtain. Any thoughts there? Would you a follow-up webinar maybe in a few quarters? Any thoughts there, Lee?
I mean,
Or
sure,
Max?
we would be very happy. We have presented at a couple of machine learning conferences, posters, and we're hoping to present another talk. We had a talk at one user group meeting, but if there's more interest, you will find us in probably at one of the conferences in our field in the near future. Yeah.
Awesome. Well, you heard it here first, attendees. Keep a lookout. We'll be sure to share Congruence's website so you can stay up to date with any updates they have, and please do follow them on social media as well. Well, that is a wrap. Thank you so much, Jeremy, Max, Lee, for your time today. I had a ton of fun. Hope you did as well. I'm sure the audience really enjoyed the conversation, and they had a ton of great questions. So thank you so much for your time, and thank you to the audience for joining us today. And until next time. Yeah, cheers.
Thank you.
Yeah.
Bye-bye.
Bye.
Bye-bye.